In this part of the chapter, we'll talk about the segmentation of Drosophila embryo according to the anterior-posterior body plan. So we mentioned that the first maternal genes are the first genes to be expressed. And if you remember, we talked about bicoid anteriorly and nanos and Oscar posteriorly. These genes are called maternal genes, and they establish the polarity of the oocyte and of the embryo according to the anterior-posterior axis. So the presence of these genes, the maternal genes, will induce the expression of other genes. And the first zygotic genes to be expressed are called gap genes. They are called gap genes. These gap genes, they occupy broad territories of this embryo, okay, or large territories, and they are called gap genes because when they are mutated, they create a kind of gap in the body plan of the Drosophila, so that different segments that should be present usually or formed in this area will be absent when we have a gap gene mutation. Anyway, when gap genes are expressed, they can induce the expression of another group of gene called pair rule genes. So pair rule genes usually they have this kind of pattern, always 7 stripes perpendicular to the anterior posterior body axis. These pair rule genes they induce the expression of segment polarity genes, and segment polarity genes are genes that will determine the polarity of each segment. So some, let's say in this segment we have the anterior and the posterior poles of each segment, and some genes will establish the anterior pole and others will establish the posterior pole of each segment. So after, when gap genes are expressed, pair rule genes and segment polarity genes, these genes altogether will activate the expression of homeotic genes in order not to form the segments themselves, because the segments are already formed, but to give the identity of each already formed segment. So these genes activate homeotic genes to give the identity of each segment. So here in this slide, I'm showing you the expression pattern of these different genes I already talked about, like hunchback gene, for example. It's a gap gene. It's one of the gap genes. It's called hunchback, and this is the domain of um, hunchback expression. You see, this is the domain in orange, and you have Krupel. It's another gap gene expressed in this domain in green, and this is the overlap in yellow between hunchback domain and Krupel domains. Okay, so in this slide, for example, you can see a Krupel. It's always expressed here in this domain, and this is the domain of expression of hunchback. This is done by in situ hybridization to see the localization of the messenger RNAs for hunchback and for Krupel, for example. You see bicoid, it's a maternal gene. It's always localized anteriorly. It induces the expression of gap genes. Gap genes induce the expression of pair rule genes. And actually we have two pair rule genes, two known pair rule genes. We have even skipped in red, always 7 stripes if you count them in red: 1, 2, 3, 4, 5, 6, and 7. 7 red stripes belonging to the even skip gene expression pattern. And another one, this pair rule gene is called Fuchi-Tarazu. It's called like this because it has been discovered by Fuchi-Tarazu, which is a Japanese biologist, okay, who discovered this pattern of this gene, and they called this gene in red Fuchi-Tarazu according to the name of the discoverer. It's a pair rule gene as well. You see that even skipped in red and Fuchi-Tarazu in black, these are expressed in every other segment. Okay, we don't have overlap between even skipped and Fuchi-Tarazu. They are expressed in every other segment to determine the segmental plan. And these pair rule genes, they activate the expression of segment polarity genes like Wnt, hedgehog, and they set the boundaries and the poles for each segment. For example, here in under electron microscopy, we can see Fuji-Tarazu expression. You see we have 7 stripes, 1, 2, 3, 4, 5, 6, and 7, which are perpendicular to the anterior-posterior body plan. So this is one of the pair rule genes, Fuji-Tarazu. And here I'm showing you engrailed, which is a segment polarity gene. Engrailed, you see, this is, for example, the segment, and engrailed is usually expressed in the posterior part of each segment. Okay, let's say we have the segment right here, for example, okay, and this is engrailed, always present at the posterior domain. Let's say this is another segment, and engrailed is always at the posterior. Other genes will be localized anteriorly. Okay, and overall we have 14 segments, 3 head segments, 3 thoracic, and 8 abdominal segments. So here I'm showing you experiments about bicoid and its role in the determination of the anterior compartments. For example, here we have a wild-type embryo. Okay, bicoid messenger RNA is located or expressed in the anterior part of this embryo, and this will determine the head, okay, in this compartment, and we get a wild-type phenotype with a head and a tail. This is a normal development. But when we have an embryo which is deficient or mutated for bicoid gene, you see that this embryo will be developed into two-tailed embryo. So we don't have, because bicoid, the anterior determinant is absent, so we don't have development of the head. Okay, so we have the formation of the telson, which is the posterior, usually the posterior part of the embryo, that will be developed as well anteriorly instead of the head. And if we make gain of function experiments, what does it mean, gain of function experiments? It means in a mutant embryo for bicoid, we add bicoid expression to the anterior domain in order to rescue the mutation or the mutant phenotype. So when we added bicoid anteriorly, we can see that we rescued the normal development and we get the head formed at the anterior compartment as if it was the case for the normal or the wild type embryo. And if we add, for example, bicoid just right in the middle, we can get the head in the middle. Okay, you see here we have telson thorax, so a tail, because we don't have bicoid. Here, of course, posteriorly we have Nanos and Oscar, which are posteriorizing factors. We get abdominal segment and telson. Okay, but here in the middle we get a head. Okay, you see H for the head. Okay, and we have the thorax. Okay, the head will be formed in the middle. And if we add, for example, bicoid posteriorly in a wild-type embryo already expressing bicoid anteriorly, we get two-headed fly. Okay, one head is normal, is at the anterior side, and one ectopic head due to the addition or the injection of bicoid posteriorly. So in this experiment, it's a normal embryo with anterior and posterior compartment. They just tried out to prick a little bit anteriorly with a needle in order to take or to let some of the anterior cytoplasm of this embryo to escape, and they inject instead, or they inject in this part after that, some posterior cytoplasm taken from a donor egg. Okay, they take, for example, from another donor egg, some of the posterior cytoplasm, okay, from this region right here, and they injected it anteriorly. Okay, so what do you expect from this experiment? So we expect that we get double posterior, so they got double posterior larval stage after some development. Okay, why? Because the posterior cytoplasm contained posterior determinant like Nanos and Oscar, and when this part of the cytoplasm has been injected anteriorly, it transformed the anterior compartment to a posterior one, That's why we have a double posterior larvae or double-tailed larvae, and the head cannot be developed because we don't have bicoid expression. Okay, this is the normal larvae and this is the experimental condition.